So today I'm going to talk about some of the below-ground consequences of mountain pine beetle. And I'm going to talk a little bit about some of our past research and then connect that to some questions that we have moving forward. So as everyone in this crowd knows, mountain pine beetles, this outbreak in Western Canada in some areas is a novel disturbance. And so that means that the predictions for forest recovery can sometimes be unclear. And so as I mentioned, we focused on the below-ground consequences of widespread tree mortality. So what's happening in soils after all these trees die, how those soil conditions change, and how those conditions might then affect seedlings establishing in those beetle-killed stands. And so when I talk about soils, I really think a lot about the living part of soils. And so I study mycorrhizal fungi. Mycorrhizal fungi, they colonize roots of most boreal tree species, and they are considered typically a mutualist. So they are taking resources from the soil, so nutrients and water, they give it to the tree in exchange for sugars that the tree produces through photosynthesis. And so it's because of this interaction that we were interested in how this widespread tree mortality might be affecting these fungi. So do they disappear? Do they change in species diversity? And we thought that they might be really hard hit by this tree mortality because, as I mentioned, they depend on a living tree for that carbon. So if you turn that carbon flow off, you expect these mycorrhizal fungi to change in abundance, change in species diversity, something is likely to happen. And so on top of that, we know that pines, as a genus, are really sensitive to the presence and the abundance of mycorrhizal fungi, and sometimes even to the species that's colonizing their roots. So this is an example, this is not from my research, but from another group, and it shows the invasion of lodgepole pine in this grasslands in New Zealand. And so before this invasion occurred, they brought our lodgepole pine into the region and they could not get it established. And it was only when they provided this ectomycorrhizal inoculum, so its fungal partner to the tree, that it was able to establish, survive, and then subsequently invade this region. So we know that pines are really sensitive to changes in mycorrhizal fungi. And so putting this together, we went into our research with this conceptual working model. So this is really simplified, but I think it pretty much tells the story. So a living, healthy pine stand. We expected, because it's allocating lots of carbon to these mycorrhizal fungi, we expected there to be lots of mycorrhizal fungi and high species diversity. So the mountain pine beetle comes through, kills a whole bunch of pine trees that dominate those stands. We expected that the mycorrhizal fungi would decrease in abundance, probably in diversity of the communities as well. And this in turn would feed back into affecting pines establishing in those beetle-killed stands. So because there's this loss of fungi, we thought this might have a negative impact on seedlings in these stands. And so we tested this model in a variety of ways through field experiments, greenhouse experiments, and we found it to be more or less true. So I'm just going to go over some of our kind of main results from those studies. So this is the first thing that we did a couple years ago is we went in and we did an above ground survey of the ectomycorrhizal fungi in pine stands close to Grand Prairie. And so this above ground survey, we looked for sporocarps. So sporocarps are the reproductive structure of these fungi. They're just the mushrooms that you see, say, in the grocery store. So we went out and did a sporocarp survey along stands that captured a gradient of tree mortality. So on the x-axis here, so these are stands that experienced high levels of tree mortality. Compared to over here, these are pine stands that were relatively intact. And so you can see from this graph that in healthy, intact pine stands, about 60% of those sporocarps are ectomycorrhizal. In these beetle-killed stands, this drops to about 10%. So we're losing these ectomycorrhizal fungi. And then we followed this survey up recently with a below-ground survey. So we used molecular techniques to identify the fungi, the fungal tissue actually occurring in the soil, and the story is very similar. So there's a strong, pronounced shift in the fungal communities in soils of these beetle-killed stands. We then wanted to focus on what was happening at the ends of these gradients. So in the undisturbed stands versus these beetle-killed stands, so the ones that really experienced a high tree mortality rate. And so what we did is we collected soils from these two different stand types, and then we brought them into the greenhouse, and we used the soils to inoculate pine seedlings growing in pots. So these soils, obviously, they're going to contain fungal propagules, probably differing based on origin, and we use those fungi to inoculate the pine seedlings. So we had 3 different treatments. We had soil collected from healthy pine stands, and we used that to inoculate pine seedlings. We had soil collected from our beetle-killed stands, and then we had a non-inoculum control. So we let the pine seedlings grow, and then this measure of performance, we were measuring the defenses of these seedlings. And this is measured by total monoterpenes. We measured them in the stems and needles. And what you can clearly see is that seedlings grown with fungal inoculum from a healthy pine stand, they have higher defenses than those seedlings grown with fungi collected from beetle-killed stands or these guys that didn't receive any fungal inoculum. So what this data suggests is that pine seedlings establishing in beetle-killed stands might have fewer defenses measured by these monoterpenes, which means that they might be susceptible to future insect pathogen attack in the future. So we also complemented our greenhouse survey or experiment with another field experiment, and we tracked the survival of seedlings. So we sowed seeds in our stands, and we tracked them for 2 years to see how they did, the seedlings established. And we found there was pretty big differences in survival of pine seedlings in these stand types. So we planted pines, or we sowed the seeds of pine, tracked them, 1% survival versus 25% survival in a healthy pine stand. And we then followed up this work because at one point we wanted to know, so what kind of fungi are colonizing roots of these seedlings? And is it the same or is it different based on these different stand types? And what we found was across our greenhouse field experiments, everything we looked at, this one common story is that pine seedlings grown with fungi from these beetle-killed stands, they're called Helotiales fungi. So there was a higher abundance of these Helotiales fungi on seedlings from beetle-killed stands than those in healthy pine stands. So these Helotiales fungi, we don't actually know very much about them. So sometimes they're mycorrhizal, sometimes they seem to be more parasitic, and their functioning seems to depend on the environmental context. So it can change, and it's context dependent. So based on that other research, so just what I told you, we had some emerging questions that came out of it that we are now following up on it. So the first question, the number one question that I've got is the most common question that people ask me is, so does fire harvesting and mountain pine beetle outbreaks result in similar changes to soil fungi? So all these disturbances kill trees in some way. But is there something special going on in a mountain pine beetle killed stand in terms of the soil fungal communities? And so this is something that we're looking at, and it's funded by the Enster Strategic. It's led by Nadir, and the co-PIs are J.C. Cahill, Suzanne Simard, and myself. And our project manager, Jonathan Kale, he will be leading surveys to address this question. So we'll sample fungal communities and soils of burn sites, beetle-killed stands, harvested sites, and then look to compare. So are they similar? Are they different? Again, trying to figure out, is there something special about beetle-killed stands, or are the fungi just responding to disturbance per se? 2 other questions that have emerged from this other research, from our previous research, was asking whether the increase in Helotiales fungi in beetle-killed stands vary by ecosite. So does this increase in these Helotiales fungi that I mentioned is across the board in all beetle-killed stands, or is it more nuanced? So does it depend on, say, moisture levels? In drier sites, they might not be there, but maybe this differs in wetter sites. And then finally, the last question we're looking at, can we offset the detrimental effects of these Helotiales fungi with inoculation? So in other words, is there a way to rescue these pine seedlings in beetle-killed stands if they are being colonized by these Helotiales fungi? Can we preempt that by inoculating them with other kinds of fungi first? And so these last 2 questions, these are being funded by FRI Research. So thank you very much for the activity team for funding this research. And so to answer these questions, okay, so for the first one, to look at the interaction of ecosite and soil fungal communities, we're going to be sampling about 40 sites spanning a range of ecosites in sort of west central Alberta. And so most of these sites have already been identified through Ella McDonald's work and the Deer Herbal Bin. So we have most of the sites. We're going to go out, sample soils, and then use molecular techniques to identify the fungi occurring in those soils. And then what we'll do, this is not obviously real data, this is just a schematic. What we'll do is we'll rank those ecosites in terms of the Helotiales abundance. So the dark colors, lots of Helotiales, down to the bottom, not so many Helotiales in the soil. And then we're going to plant pine seedlings into the ends of those gradients. And those pine seedlings will have one of 3 different treatments. So either they will receive soil inoculum from healthy pine stands, or they'll receive inoculum, fungal inoculum from a beetle-killed stand, or they won't receive any inoculum. So we'll inoculate the seedlings, we'll put them out in the field, we'll let them grow for a couple years, and then we'll measure their performance in terms of growth, survival, and then also look at their defenses in the field, in a field situation. So together, these new projects, I think there are so several key deliverables about these new projects. So first, we'll be able to do an assessment of soil health in beetle-killed stands, which might be useful for some kind of triage approach. So maybe, these stands that have lots of Helotiales fungi, and then the transplanted seedlings really don't do so well on them. Maybe those are stands that we want to focus on for rehabilitation. This research will also directly test whether fungal inoculation is beneficial. So is this actually some kind of management strategy we want to employ? Is there any benefits to it? Does it matter? There might be situations that it might not improve seedling growth. And then finally, what I think is really important, it will put changes in the soil fungal community in the context of other prevalent disturbances on the landscape, so like harvesting fire. So again, trying to get to this question, is there something special in beetle-killed stands, or are the fungi just doing what they always do after a disturbance? So finally, just some acknowledgments. Again, so thank you so much to FRI Research for supporting our upcoming research, to NSRIC and Alberta Conservation Association. They've been really good at supporting graduate students in some of their research. So thank you very much.